Okay, so we're going to talk about translation. So this is the last step in the central dogma of molecular biology. So remember, we started out with double-stranded DNA. That double-stranded DNA, and this lives in the nucleus. And then the first thing that happens is the double-stranded RNA gets transcribed, a process called transcription, into single-stranded messenger, so SS, single-stranded messenger RNA. This process all happens in the nucleus. So I'm going to draw a little, let's pretend this is the nucleus. So everything in this corner up here, this is all the nucleus. And then outside here would be the cytosol. Except right outside the nucleus, there are all kinds of what's called endoplasmic reticulum, which is back and forth, all over. There's lots of good pictures of this in your slides and in the videos, the different videos that I posted about transcription and translation. And on the endoplasmic reticulum are ribosomes. And when we have ribosomes on the endoplasmic reticulum, it's called rough endoplasmic reticulum. What makes it rough is that it is studded with these ribosomes. Ribosomes is the site of protein synthesis. Protein synthesis. The process of protein synthesis from a transcript is translation. And that's what we're talking about right now. So we talked about transcription in the last video, going from double-stranded DNA to single-stranded messenger RNA. The next thing that needs to happen is this single-stranded messenger RNA is going to leave the nucleus through what's called a nuclear pore, which only messenger RNA can go through. Double-stranded DNA cannot go through the nuclear pore. So this single-stranded messenger RNA is going to go through the nuclear pore, and it's going to find a ribosome. On the ribosome is where the messenger RNA is read and translated into protein. So I'll write that. Messenger RNA leaves nucleus through nuclear pore. Messenger RNA finds ribosome. Remember, ribosomes are made up of rRNA, ribosomal rRNA. The messenger RNA finds a ribosome. The ribosome is made up of two subunits. And if you're dealing with bacteria, prokaryotic organisms, or eukaryotic organisms, they're different. But it essentially kind of makes a tunnel through the middle where this transcript is going to kind of thread its way through. So the messenger RNA finds a ribosome. Messenger RNA is translated into protein on the ribosome. It's translated into protein by the tRNA bringing the appropriate amino acid to the growing protein. Okay, so this is all kind of a little abstract. I am going to write a code and then we'll go through it. Okay, so let's pretend This is my messenger RNA. Transcript. And this messenger RNA, remember, so it doesn't use T, thymines, it uses U, uracil. So I'm going to say, here's my code. A, U, G, G, U, U, G, G, C, G, A, U. And this is the beginning of the code that is also on slide 50. So what happens is this is all being threaded through the ribosome. So this is, it's got like, so it's like got two kind of subunits. And then the messenger RNA is going to kind of thread its way through. And as it does, it gets read. It gets read in groups of 3. So we'll say ribosome reads messenger RNA in groups of 3. These groups of 3 are called codons. There is a codon chart on slide 52 of this last lecture, and it shows how each codon, there's a 3-letter code for each amino acid. So let's just start at the beginning. So here's AUG. This is the first amino acid of every single protein. It is methionine, but it's a special kind of methionine. It's called formylated methionine. It's called F-met. Every single protein starts with F-met, and then it will get used later. Other methionines are coded for with not AUG. There's other 3-letter codes for methionine. Only AUG, this is only for the first amino acid. Okay, every protein starts with AUG. So then the next one, if you look at the codon chart, GUU. So if we find GUU, we see that this codes for a valine. And then GGC. If we look at the code on slide 52, we see GGC. GGC is glycine. And then GAU is aspartate, or aspartic acid. So this is how we read the transcript. So we read the transcript, and now this is going to be the amino acid sequence. And these it usually shows this, sometimes coming out the top or the bottom, that as this gets read, we would have F-met, valine, glycine, aspartate. We're having this growing chain of amino acids. I did this backwards. It should be this way. F-met at the top, then valine, then glycine, then aspartate, because it's growing this way. So the next amino acid would grow here. This whole thing would bump out 1. This is something I always show a cartoon video of in class, showing these things physically moving around because it can be really abstract. Slide number 50 shows this happening step by step. So the messenger RNA is translated into protein on the ribosome. The tRNA brings the appropriate amino acid to the growing protein. So the ribosome reads this and it says AUG, that's F-met. So that means a tRNA is going to bring the F-met there. tRNA stands for transfer RNA. And what it is because there are 20 amino acids, but actually there's way more. There's 64 3-letter codes that code for amino acids, which means every 3-letter code that is on this chart on 52, there is what's called an amino acyl tRNA for each 3-letter code. So as this transcript is being read, whatever the 3-letter code is, the corresponding amino acyl tRNA is going to bring that amino acid to the ribosome. The codon is on the messenger RNA. The tRNA, let's use a different color, which brings the correct amino acid, has what's called the anticodon. So the way that this tRNA brings the next correct amino acid is by binding the complement of the codon. Codon is the complement to the anticodon. There is a picture of the structure of a tRNA in your lecture notes and in your textbook. It looks kind of like a neighborhood with cul-de-sacs at the end. And down here, there are 3, 1, 2, 3. These 3 places, this is the anticodon. And this is what's going to bind to the messenger RNA in the ribosome, and attached to this would be whatever amino acid. So for example, the anticodon that would bind here for FMET, for methionine, would be the complement to AUG. So the complement to A is U, the complement to U is A, and the complement to G is C. So what is on the anticodon is the complement to the codon. That's how you would know which one goes here. And the amino acid that would correlate would be the formylated methionine here. So then the anticodon for valine here would be CAA. The anticodon for glycine would be CCG. The anticodon for asparagine would be CUA. So remember, it's the complement, back when we learned about the nitrogenous bases, and you see that every nucleotide has a complement. So if it's A, its complement is U. If it's U, its complement is A. G, C, C, G. And remember, these are all RNA. The tRNA is an RNA. The messenger RNA is an RNA. So we're all going to have U's here, no T's. So translation also has 3 stages like transcription. Initiation, which is the FMET, is always the first residue. Elongation, which this process will continue. So there's initiation, elongation, and termination. So initiation, this is the AUG, FMET. Elongation goes along, and this grows one by one. Something that's important to note here is that we said that enzymes that are catalyzing all these chemical reactions are proteins. They're proteins with enzymatic activity, catalytic activity. All enzymes are protein, except the ribosome acts like an enzyme here. It catalyzes the transfer of these amino acids, the growing polypeptide chain. It's called a peptidyl transferase. ACE, it's an enzyme. This is the only RNA. It's the only enzyme that's not a protein. It's an rRNA enzyme. This is the only example of an enzyme that is not a protein, is the peptidyltransferase, which is the rRNA during protein synthesis. This will continue reading this code until it reaches what's called a stop codon. And what's important about the stop codon is that there are anticodons, the tRNA, instead of bringing the next amino acid, it would bring what's called a termination factor, which would cause the protein synthesis to stop. So again, this is happening on the ribosome, and there's 3 sites in the ribosome where this is happening. There's the A, arrival site. There's the P, the peptidyltransferase site, and then there's the E, the exit site, and it's going in this direction. So it arrives, it goes to the peptidyltransferase site, and then it exits. Let's see what else to say. Definitely watch the videos and read the text for this. There's not a really good way to draw this out. It's much easier to see in the lecture slides and also in the cartoon videos that I posted. There is a slide on slide number 74 which shows this whole thing. The difference between what's on slide 74 and here is this, you would know that this is a eukaryotic cell because there's a membrane-bound nucleus. That's what this line here is representing. On slide number 74, that would be a prokaryotic cell because there's no nucleus. If there's no nucleus, this whole thing is just happening continuously, all next to each other. The DNA next to the ribosome, and all of it is happening in one slide. So you'll see all of that on slide number 74. Another important thing to note is that the size of these ribosomes are different in eukaryotic cells versus prokaryotic cells. And many antibiotics that we use to kill bacteria target protein synthesis in bacteria because the antibiotics that do this, they exploit the difference in protein synthesis in a prokaryotic cell versus protein synthesis in a eukaryotic cell. So these are very effective. These would be called broad spectrum antibiotics because they would inhibit protein synthesis in all bacteria. So they wouldn't be killing just a certain type of bacteria, they would be killing all bacteria because bacteria use different size ribosomes than we do. So this is a good way to kill bacteria. Even though it's non-specific, it would kill all bacteria. But if you're having a strong bacterial infection, sometimes a broad spectrum antibiotic is the best choice. And it does that by inhibiting protein synthesis. Again, I'll post I'll post some worksheets to go through examples of using this codon chart on slide 52, where I'll give you probably a DNA sequence. From the DNA sequence, you could transcribe the messenger RNA. From the messenger RNA, you could break it up into groups of 3 codons and use the codon chart to translate it into a protein sequence. So definitely look at the slides, look at the other videos and the worksheets to kind of wrap all of this up on protein synthesis and translation.